Hi, my name is Jacqueline Phan, and I'm a PhD student studying under the advisement of Dr. Ernesto Abel Santos in the Department of Chemistry and Biochemistry. Now, you may be wondering why the title of my talk is Poop. There it is. Well, that's because my research focuses on a major problem in our healthcare setting called Clostridioides difficile infection, or CDI, which can cause severe diarrhea. We have actually discovered a biosalt analog compound that can protect mice and hamsters against this disease. And hopefully by using this research, we can then develop a way to protect humans against this disease as well and help them get their poop back to normal. CDI is caused by rod-shaped bacteria called Clostridioides difficile, or also known as C. diff. A key characteristic of C. diff is that it is able to form these tough and round structures called spores that act as the infectious form of the disease. Spores are basically like super eggs that can survive in many different environments until a trigger causes them to hatch, or what we call germinate, into toxin-producing cells that can then eat away at the intestines. This germination process is required for CDI symptoms to occur, and it just so happens that the gut can be a perfect place for these spores to germinate in once the right conditions are met. CDI can cause abdominal pain, nausea, vomiting, explosive diarrhea, gastrointestinal bleeding, and in worst case scenarios, even death. Patients who are elderly or immunocompromised or those who have recently taken antibiotics are much more susceptible to contracting CDI. So if we are not part of these populations, why should we care about this disease? Well, here are 3 reasons. The first is that CDI is a very serious problem. There are over 200,000 diagnoses per year in just the United States alone, and up to 25% reoccurrence within just 2 months of diagnosis. The second reason is that CDI is a very deadly problem. There are over 12,000 deaths per year within just 30 days of diagnosis. The last reason is that CDI is a very expensive problem. It can cost an average of $35,000 to treat a single inpatient CDI case, which can then add up to about $3 to $5 billion in annual cost burden to the U.S. healthcare system. Luckily, our research focuses on how biosalts that are naturally produced in our liver and secreted into our intestines have potential in the preventative treatment of CDI. In our bodies, there are some biosalts that act as natural germinants of C. diff spores, while other biosalts act as natural inhibitors of spore germination. In healthy individuals, the natural gut bacteria actually help regulate these biosalts. So as we go further down the gut, there is less natural germinant and more natural inhibitor. Therefore, the spores that enter the body will exit the body as spores and not cause CDI symptoms. However, in patients who are immunocompromised, that natural gut bacteria is depleted and is much less efficient at regulating these bile salts. So as we go further down the gut, there is more natural germinant and less natural inhibitor. So the spores that enter the body will germinate into those toxin-producing cells that can then cause CDI symptoms. Our research aims at producing a synthetic biosalt analog compound that is a better germination inhibitor than the natural inhibitor in our bodies. After screening hundreds and hundreds of new compounds, we finally came upon one that we called kappa that was able to prevent spore germination over 100 times better than the natural inhibitor. And unlike its predecessors, it actually prevented spore germination with various strains of C. diff, including a hypervirulent strain, which means that it can cause severe disease. We tested kappa as well as other synthetic biosalt analog compounds in 2 ways. The first way was called an in vitro study, which means in plates. We used something called a germination assay, which could tell us whether a compound was effective against preventing spore germination in plates. We took C. difficile spores and we incubated them with their natural germinants, taurochole and glycine. And we also incubated them with the synthetic bile salt analog inhibitor compound that we were testing in various increasing concentrations. We put them into a 96 well plate, and then we put that plate into a special plate reader, which gave us a reading called optical density. Now, optical density is a very effective method at measuring spore germination because spores are optically dense structures and they can lose their optical density over time if they germinate. So if a compound is effective against preventing spore germination, it will remain at an optical density of 1 over time, just like we see with this gray line representing no germination. However, if germination is occurring, meaning that the compound is not as effective at preventing spore germination, then we will see a reduction of that optical density over time, as we can see with this blue line representing germination. Kappa at very low concentrations was actually able to prevent spore germination of various strains of C. diff. Now, there's a saying in science that what happens in vitro stays in vitro. So now we must test kappa in another study called an in vivo study, which means in live models. We use 2 types of live models. The first is a mouse study and the second is a hamster study. Both are important to study when looking at CDI. The mice study is a newer model. They are resistant to CDI, which means they need many spores to even get them anywhere from mild to severe signs of CDI. They also have a slower clinical endpoint, which means it takes them longer to get to the point where we can see what's happening to them. And then they also don't have any relapse model, which means that they don't tend to get sick again after initially getting sick. However, hamsters, which is the traditional model, they are super highly susceptible to CDI, and only a few spores can actually cause them to reach their clinical endpoint and get severe CDI symptoms. They also can relapse from CDI after initially getting it the first time. Now, why is it important to study both of these models? Well, because humans actually lie somewhere in the middle. We start with our mice model, where we give the mice antibiotic cocktail water to help deplete their natural gut bacteria. We then give them the C. difficile spores and also the compound we are testing, in this case, kappa. We then see whether or not the animal is sick from CDI or if they are protected. We use a special CDI scoring rubric where we take the symptoms and signs on the left side and we tally them together to determine whether or not the animal is non-diseased, has mild CDI, moderate CDI, or severe CDI. And if they do have severe CDI, we euthanize them right away so that they do not have to suffer. We see that when mice are not given kappa, they actually succumb to disease within 48 hours after infection. Some mice get even very severe CDI to where they have to be euthanized. However, mice that are given that kappa treatment actually appear to be non-diseased. And when we actually give other more dangerous strains of C. diff to the mice and treat them with kappa, we either see that they look non-diseased, have reduced CDI, or even delayed CDI symptoms. Now we have to see whether or not our compound is effective in the very susceptible hamster CDI model. We take hamsters and we give them a shot of an antibiotic called clindamycin, which helps deplete their gut bacteria. Now, because hamsters are very sensitive to changes in their gut, we also have to give them small dosages of another antibiotic called vancomycin, which actually helps protect them from some of the dangerous effects of clindamycin. We also give them a bile salt analog compound to help protect them when they do get C. difficile spores. And then we check whether or not they do get CDI or whether or not they are protected. What we see in the hamster model is a little bit different than the mice model because hamsters are so sensitive to C. diff spores. we actually look at something called our survival model, and we actually see that with kappa, that animals have a higher chance of survival than those who do not have kappa. For all animals that did not get kappa and did get the C. diff spores, they all succumbed to disease by day 8, whereas those who had kappa had an 80% chance of survival. Therefore, kappa appears to be a very promising compound in our future studies with these biocell analogs. Now, C. diff in the past has been traditionally associated with those who are elderly, but in more recent years, it has also been associated with many others in the community. It is important that we continue to study these biocell analogs to help continue to protect all members of our community. We hope that by continuing to do this, that we will be able to help patients say again, poop, there it is, and see their normal poop once again. Thank you for listening, and I would like to acknowledge my fellow lab members as well as all those who contributed to this research here, including our funding from NIH and the Nevada Space Grant. Thank you for listening, and please let me know if you have any questions. Have a nice day.